Well, thank you, Angel. It's my pleasure to be here, and for more than one reason. I checked this morning. The temperature in Brookings, South Dakota, was a crisp minus 17 Celsius. Last week at this time, it was minus 35 Celsius, and the wind chill was minus 50 Celsius. So I appreciate the fact that it's actually above zero in terms of Celsius here. So thank you. So what we're going to do today then is, as Angel said, is actually talk about the connection between immunology and particularly the gut. And I'm really going to spend a lot of time talking about that today. I first want to give credit to a number of people whose slides I have either borrowed or absconded, but most of them actually gave them to me willingly. So, but in particular, Mike Kogut and Lance Baumgard. You'll see several things from those guys, and then a few other folks as well. So we're going to talk about what I call the 3 levels of immune protection. And those 3 levels are going to be the mucosa, the barrier itself, the innate immune response, which I term as sort of the good, the bad, but we're going to talk particularly about the ugly that occurs with that, and then the acquired immune response. But we'll spend most of our time actually on these first 2 today, because I think that really relates well in terms of what we're talking about in terms of the use of prebiotics and probiotics. So let me start out with actually a slide from Mike Kogut, who's just down the road at the ARS facility at College Station, and this is his question: what is gut health? And gut health is any way you want to define it. What he puts up there for those of us that are from the U.S. with a little historical background is the fact that back in 1964, there was a famous case determined about obscenity in terms of pornographic materials, and in this chief, one of the justices, their answer to what it was is, I know it when I see it. And that's sort of about gut health, is that we're going to have different definitions for actually what gut health is. Well, let's just talk a few things about the gut. So in particular as it relates to immunity. So again, it's the largest interaction between the external and the internal host milieu that we have. Again, we know it's the major barrier, but the thing that I want to leave you with today, it is by far the largest immune organ of the body. By far, the largest immune organ of the body. And so it acts as a physical, but also as an immunological barrier to pathogens. And we'll talk about that particularly today. And then on the other side of that, and this is this dynamic that occurs, is on the other side is this natural habitat of microbes, the microbiome, and the interaction that they have in between the two, between the immune system that is the gut and the microbiome is huge in terms of how that regulates both our gut and systemic functions, and particularly as it relates to immunity. So again, if we think about, again, the components of this healthy GI tract, and again, the thing that we think about often when we're talking about from a nutritional standpoint is this whole thing about digestive and food absorption. And I'm going to not spend any time talking about that at all today, but rather the other things that are listed up there. So again, this idea of having this stable microbial population is absolutely essential to maintain the immune integrity and the barrier function of this organ. And then we'll talk again quite a bit about, obviously, the immune status. There's a lot of things that we're beginning to understand. I mean, there's direct connections between the gut and the nervous system and the immune system that occur right in the gut. And then, again, we'll spend a lot of time talking about this gut barrier, and we'll talk a little bit in particular about the neuroendocrine system. And this really relates particularly as we talk about stress. So the effect that stress has, and we'll define stress here in a little bit, but the effect that stress has on the digestive system and also on the microbial system itself. So let's begin talking a little bit about this. So we have this, what I call the neuroimmune interaction. And it depends on who you talk to. You'll find that that if you talk to Mark Light at Iowa State, he will tell you that in fact what drives everything in the gut, it's not the animal, it's not the neural system. In fact, it's bacteria. That bacteria themselves are the driving force of the immune response in the gut. But if we look at, here's the gut, and the idea that we've got this microbiota, okay, that are present, that are sending signals continually to the immune system, and hopefully what most of those signals are going to do is they're going to help in terms of maintaining health. But we know that sometimes that those signals are actually going to result in infection, and then we know there's other things that can be present, like toxins. But then we know in terms of positive things that those microbes are in terms of producing vitamins and other metabolites that are essential for that animal's lean growth, and then of course in the actual absorption of materials as well. So all this is going on, and there's this interaction continually going on in the gut and in between the immune system. And then I want you to realize that it's also all tied to the central nervous system as well. Okay, so this is a slide that I got from someone at NIH, because I think it's sort of a good way to think about this whole idea of this interaction. So if you think of the body as this hollow plastic tube, so this is what we call PVC pipe, okay, and so the food's digested within that hole, but it never actually enters into the plastic. And we know then that that tube itself is the digestive system. The inner surface of that is the digestive system. The outer is the skin, and the only part that's actually the body is this part right here. So basically we are surrounded by microbes, and the animals are surrounded by microbes both internally and externally, and the immune system has to sort through that. And it has to figure out, what's the good guys, what are the things that I need to do, what are the cues that I need to pick up on. So you're going to see this particular diagram a few more times today, because it's sort of my fundamental thing to help explain these three levels that we're going to talk about today. So again, the first thing that we're going to talk about is we're going to spend time on the barrier, and we'll talk about that in detail, but I just want to walk you through, for example, let's just take the example that we're going to give a vaccine. So with a vaccine, what we're going to do is we're immediately actually going to go past this barrier. Because either we're going to give an injection, or we're going to give it to them orally or intranasally, but we're going to do it in such a way that we bypass the barrier, and we do that on purpose, because we want to then turn on the immune system, and what we turn on then is this innate immune response first. And I think of the innate immune system kind of like, think of it like a smoke alarm. Its job is to sense what's going on, and then when it senses something's going on, it sends us signals then to respond to that. And it does that all the time, okay? Now the thing about it doesn't have any memory, okay? So just if there's a cut, the innate immune system goes after it. If you give a vaccine, the innate immune system responds to that. If it's a hot heat stress day, the innate immune system responds to that. So its job is to respond to these stimuli. So it could be pathogens, it could be environment, and often when we talk about stress, this is the system that's turned on, okay? So it's in terms of the innate immune response. And it consists really, there's two parts to it. And that is there's a cellular part, okay, and there we talk about particularly about things like neutrophils and macrophages whose job is to eat things, is to phagocytize, is to take things up and eat them and destroy them, and at the same time send signals. And on the other side is what we call the humoral factors, and that's particularly what we call these things that are called cytokines. And in particular, what's important with innate immunity is its ability to turn on inflammatory cytokines. So its job is to turn on inflammation, because what inflammation does, inflammation helps recruit cells. It increases blood flow into an area. It sends signals, as we'll see, to other parts of the body to say, hey, I need some help here. But again, inflammation then is a key. And if I'm given a vaccine, for example, and particularly if I'm given an injection, a parental vaccine, and if I give that vaccine the first time I give that, and those animals have no indication of all of inflammation. So I don't see a droopy ear, or I don't see animals off feed, or I don't see some other things that would indicate to me that innate immune response has been turned on, I would contend that that vaccine is probably not going to work very well, because for any vaccine to work, the innate immune system has to be activated. And if the innate immune system is not activated, the adaptive response won't be turned on. Now, the adaptive response is a response that we think about when we think about vaccines, because this is where memory is. So if I give a BVD vaccine, and I'm going to vaccinate that animal, I'm going to get T cells and B cells that will be there hopefully for the rest of that animal's life. And that's what we call memory, okay? But memory and adaptive immunity doesn't occur unless first I turn on innate immunity. These two things work hand in glove. And so one of the things that when H and I, in a former life, we'd be dealing with veterinarians or producers, and they'd say, I want a vaccine that's so smooth that I didn't even know that it affected the animal. And I would contend if it's so smooth that it didn't affect the animal, it didn't do anything. So if you don't see anything, nothing happened, okay? Now, as you revaccinate animals, that becomes less of an issue, or less likely to occur. But when we turn on a vaccine, we give it that first time to an animal, and particularly as an injection, we should expect that some of those animals are going to have some side effects. And that side effect may be going off feed, it may be some sweats, it may be some other things. But what it's telling us then is the vaccine's working. The vaccine got the immune system's attention. Okay, so let's talk a little bit more about stress then. Okay, so
one of the big definition of stress is anything that affects the immune system, okay? You can see I've got this is a short list up here of things that we know that indeed affect the ability of the immune system to respond. So things like commingling, injury, water. Water is a huge thing when it comes to the immune response. So John Richardson, who's here at West Texas, did a great study back when he was in Arkansas, and partially here as well, where he looked at animals after arrival. And these were 6 to 8 week cattle, brought them in 24 hours after they were in the yard. He pulled a blood sample from each of those animals, and he looked at what we call the CBC, or just the regular blood parameters. So the red blood cells or white blood cells. And the interesting thing that he saw, that one of the highest risk factors was about 4 times more likely that if cattle were dehydrated, and this is 24 hours after they've been in the yard, they are 4 times more likely to have bovine respiratory disease. And that's because, among other things, water is essential for the immune system in at least 2 different regards. One is we're going to talk about the barrier. And the other thing is these cells have to move from point A to point B. And as soon as an animal is dehydrated, it completely changes the dynamics of how cells can move. And if they can't move, they can't get where they need to go. So dehydration is a huge issue. And we'll find, and I've seen, interesting, I've got a couple of presentations lately into some calf raisers, and the interesting thing is that when you talk to people that are very, very diligent about making sure that young calves get water and ask them about how much BRD or how much problems they have with respiratory disease, they will tell you there's a dramatic decrease if cattle have, even young calves have water, continually have water. All right, then we're talking about feed, and here we're talking about just in terms of energy being one of those things, but also in terms of the microbiome, and we'll talk about that later, because what we feed animals influences the microbiome that they're going to have. And then we've got things that have to do with stressors like density and pen number. And then heat stress is far and away of the environmental stresses the most severe stress on animals. The innate immune system is activated in a way that is not good when it comes because of heat stress. Okay, so again, trying to avoid, again, if we're talking about our vaccination example, trying to avoid vaccinating animals on hot days, or certainly in the heart of the hottest part of the day, is important. But again, heat stress really has a major effect on immune response. And we'll talk more about that later, particularly in regards to some things that Lance Baumgart has thought about over the times. Then we've got things like handling and movements, and fear and flight. And we'll talk about this, but fear and flight used to be, that was our definition of stress. That if we somehow startled the animals, that was stress. So there's a lot of other stresses that occur before that, but the interesting thing about fear and flight is there we're talking about norepinephrine and epinephrine. And the interesting thing about norepinephrine, again, this is some work from Mark Light, has shown that clearly that a number of gram-negative bacteria actually grow better when norepinephrine is around. There's actually receptors on the surface of the bacteria that say, hey, this animal is stressed, let's grow to beat the band. So if I'm Mannheimia or I'm Salmonella, I will grow better when that calf is stressed, because those bacteria sense norepinephrine and use that as a growth factor.
Chris,
Yes?
I forgot to tell you, we have people from 9 different countries. Slow down.
Okay, all right. Okay. And you know that's hard for me to, because I don't like this, as if you can't tell that. So anyway, yeah. So anyway, weather extremes. We're talking again about cold and heat. So for example, I told you last Wednesday it was minus 35 in Brookings, South Dakota, and on Saturday it was plus 5. So in 48 hours we made that big of a switch, and those extremes are very difficult on animals. Then we know that we have things like certainly dust, and then competition. That gets us back again to this idea of density and pen number, because again affects how many animals get to the bunk, and also in terms of their intakes. And then the last thing that I have up there, which people might say, well, vaccination is a good thing. It's true, it's a good thing if we give it to animals that are at the right stage of the game. But if we give it to animals that are under a lot of stress, it itself is a very large stressor. Okay? So not just from the fact that we have to move animals to vaccinate them, but the vaccines themselves in animals that are already stressed will actually sometimes push them over the top. The animals will get sicker because they were vaccinated. So it's, again, it's key to make sure that when we vaccinate, they're trying to do that at times when we've tried to minimize these other things that are up there. These cattle are not dehydrated. They've got good intakes already. It wasn't just coming off the truck, okay? So, and those kind of things. So those things are all important in terms of trying to enhance our vaccine response. All right, so as I mentioned in the previous slide, this idea again that for example, fear and flight has an effect in terms of turning on these particularly norepinephrine, and that those have an effect then on each other, as you can see here. So if you start having an infection here, that affects the immune system. Okay, so again, back to the idea that let's say it's a gram-negative, then you have the autonomic nerves kicking in, that affects the bacteria, affects the immune system, and you can see all these arrows go in both directions. So again, the idea that if I have an animal up here, so that if it starts out as a physiological stress, okay, so it's a heat stress or whatever it is, that stress affects the central nervous system, which then in turn affects how the immune system is going to interact, and then we end up, again, these things going in both directions. So you need to, again, think about this is a very dynamic system. And so being able to try to minimize the different stressors that we have, and so whether it's transportation stress or the things that we see on this side in terms of how we handle animals is very important, again, to try to minimize how those animals are going to respond and how their health is going to be affected. All right, so as I told you, we're going to see this particular diagram a couple times. So here, let's look at it again. So now we're going to talk about this as what I'm calling the mucosal immune response. So we're going to particularly talk about this barrier and look at the barrier in more detail. So we're going to look at the profile of that barrier, and then we'll look at the other components, because then all these components are also part of the gut, okay, in terms of, so they're part of, so the barrier is the first thing we're going to talk about. You'll see, but see these other pieces are going to be there as well. So it's all kind of integrated, but we're going to really spend our time initially looking at the barrier, and then we'll talk about innate immunity and then adaptive immunity. Okay, so if we look at this idea then that the mucosa is that first line of defense, I want to really talk to you about sort of what I think are at least sort of 4 main areas that we need to think about. So the first is mucus, okay? So the first thing is just the presence of mucus. And the interesting thing about mucus, and we're only really beginning to understand this, and most of the work has been done in the monogastric, but there's, depending on where you're at, you have different thicknesses and different types of mucus, okay? And that has a big effect in terms of the health of the gut mucosa. So that's the first thing, and that's produced by goblet cells. So that's produced by goblet cells. So the mucus is the first thing. The second thing, and this one is a little bit harder for us to get our arms around, and a lot of the work that's been done with that has been done looking at particularly messenger RNA, but that's something called antimicrobial proteins. So I want you to think about these kind of like the body's natural antibiotics. So the body produces these antimicrobial peptides in response either to stimuli that they get from the microbiome or from the immune system. And I'll show you that in a movie here in just a minute, because that's the second thing, antimicrobial peptides. So these are, again, proteins produced by these cells that have an antimicrobial activity. The third thing, and this is not produced by these cells, but exported by these cells, and that's IgA or IgG. But in the gut, we're talking about particularly IgA. So we're talking about an immunoglobulin that's produced by the adaptive immune system that's specific for whether it's Salmonella, or E. coli, rotavirus, but it's produced by B cells that are here, but it has to be exported by these epithelial cells. So the health of these epithelial cells is essential then for the export of immunoglobulin. And then the fourth thing are the tight junctions. And interestingly enough, the tight junctions, which knit these cells together, so they knit them together to again help reinforce this barrier, they are regulated by the immune system. So if we have inflammation, and I'll show you this in a little bit, the tight junctions will break down. Okay, so we want to try to minimize inflammation so that indeed these tight junctions remain in effect and help us in terms of maintaining that good gut barrier. And so the last thing you see, my point again here about dehydration again, is if we have decreased water intake, it's going to have a direct effect on the function of this barrier. You may have to advance for me. I think my batteries are dying on me. Okay. So we just put this in terms of this idea of what I call this the kill zone. So if you take the mucus plus these antimicrobial peptides plus this immunoglobulin, it gives us a zone then in which it's difficult then hopefully for bacteria or for viruses to exist, and helps protect that mucosa then. So, and I call that then the kill zone. So it's consistent of that mucus, that antimicrobial peptides, and that IgA all together in that. And we know, interestingly enough, that the thickness of mucus will actually regulate the amount of IgA that might be produced. It will also have an effect on the antimicrobial peptides. So again, that idea about how important hydration is, I can't state that too much.
Chris, if you had more mucus, is that better or worse?
Yeah, so in most cases, more mucus is better. Okay, more mucus is better. And it varies, Ross, in terms of where kind of where you're at looking at the gut. But in general, that would be true. Okay, that more is better. So it turns out the things that I've described to you in terms of what occurs in the GI tract are also pretty true for the respiratory tract, and that's because the cells that are there are very similar. They're a columnar epithelial cell. Whereas if we look at the reproductive tract and the skin, they certainly have the immune system has a good function in them, but it's a little bit different because there we're talking about a much flatter squamous cell. It's a little bit different in terms of how that response would be. Okay, so what we're going to look at now is we're going to look at a movie that's going to kind of put this all together. And I have a really good guy on the computer now. That's good. Okay, so what we're seeing here is this is actually a movie that talks about Crohn's disease in people. So I'm going to be the narration for this. And so what we see here is, again, so here's our epithelium. And here you see goblet cells whose job is going to be to produce mucus. So here you see the lumen of the gut where the microbiome is, and this is the immune side. So again, and then these are called parietal cells. In this particular model, they're the ones that produce the antimicrobial peptides, but in cattle and in swine, basically all these cells produce antimicrobial peptides. So here you see again the production of mucus. So again, that's sort of the first part of that kill zone that we talked about. So you see that being formed, and again, really dependent on hydration and also the area of the gut that we're looking at. Then you see the production of these antimicrobial peptides, and we'll look at this a little bit more in just a second in terms of how those are produced, and then you see the tight junctions. So, and then the fourth component is IgA, and we'll see that actually produced here in just a minute. So, so while this is going on, the immune system is actually monitoring what's going on. This is sort of fascinating. The immune system can actually extend processes up into the gut and actually sample what's in the gut, and then give signals then to the immune system about what it wants to do. And so the interesting thing is there's T cells here. Okay, so that's part of the adaptive immune response. And what the gut wants to do is it wants to regulate these cells so they say to the immune system, don't over-respond. And we call that a regulatory cell. And that is driven by, as we'll talk about here, that's actually driven by what's in the lumen itself. And then what you see, it affects then B cells, and then they reproduce IgA, and then that's exported. Okay, so that's how we get IgA to the surface. So again, not produced by these cells, but it is, those cells' health is necessary. Now what you're going to see is you're going to see these cells send signals now to another innate immune cell, and it's going to send signals to say, make more of those antimicrobial peptides. Okay, so make more of the peptides. Okay, and that's what it's doing there. Okay, and so the immune system is doing this all the time. It's looking to see what's out there, and what the gut wants to be is what we call anti-inflammatory. It wants to regulate itself so it doesn't over-respond to what's going on out in the gut or in the skin or any place else where it interacts. So it does that by definition. Now what they're going to show you is that within these cells, in terms of there's a sensor that actually can sense bacterial products and say, if they're there, I need to make more antimicrobial peptides. So in this case, they're sensing something called myramyl dipeptide, which is made by salmonella. And when they sense that, then they make more of that antimicrobial peptide. increase the amount then of antimicrobial peptides out there, and keep that kill zone intact, okay? Now, what they're going to show us in just a second is that some people that have Crohn's disease have a defect in that receptor. Okay, they have a defect in that receptor, and they can't detect those bacterial products. So we'll see them zero in on this again. And now what you're going to see is that receptor has a kink in it, okay? It has a little kink in it, so now it doesn't recognize those antimicrobial peptides, and so instead of going in and making signals now, you'll see that actually that kill zone is going to decrease. Okay, so it's going to decrease. And then we'll watch what happens when that occurs. Okay, so now the kill zone is going to decrease. I have less antimicrobial peptides there, and now the bacteria have the ability to cross over. And as they cross over, what's going to get turned on? The innate immune system, because remember, its job is to be like a fire alarm and sense if something's going on, and that's what it does. So it senses that something's going on, and now all hell breaks loose, because the innate immune system brings in lots of cells whose job is to come in there and clean this mess up. And when it does that, one is it expends energy, and the other thing is it causes damage. And we'll talk about that a little bit later. Again, what we want to do is try to minimize. We need this present. We just need to try to keep this just so that it's behaving correctly and not incorrectly. So things like Johne's is incorrect. Crohn's disease is incorrect. Okay, in terms of the immune system is over-responding, and oftentimes a lot of gastroenteritis is the immune system overreacting. Okay, so now we've talked about the mucosa itself. Now let's look at the other side of the equation. Okay, let's look at what's out in the lumen, the microbiota. So we know that that is the largest collection of organisms in the body, and it's so big that in fact we can even refer to it as a superorganism, because there's actually these are communities, and they work together. And this is based on a monogastric, not in a ruminant. But we know that if you look at the number of organisms that are in a monogastric, it's about 10 to the 14th. Well, but 10 to the 12th of them are good guys. They're commensals. They're there. They're necessary. They're very useful in terms of the immune system. And we know that, and we know this. So, for example, at South Dakota State, we have ntobiotic units. That's where you have germ-free animals. And if you look at their immune systems, they never develop unless we expose them to bacteria or to some other kind of ligands. Their immune system will never develop, okay? And so when you ever heard of the bubble boy or those kind of things, the reason they had to keep them in there is because their immune system could not function well enough because they were missing the immune system, and so they were totally susceptible. And we do that sort of in reverse by taking quote-unquote normal animals, put them in a bubble, and their immune system won't even develop. So again, they're essential for immune development. We know that. And we know that the composition is influenced by the host's immune status. So the intake of that animal is going to have, whether it's on high roughage or on concentrates, is going to affect what it looks like, the health of the animal, the age of the animal. All those things are going to influence these bacteria or this microbiome, which then in turn is going to affect the immune system of that animal. So this little schematic just kind of puts that together. The idea then that the microbiota is going to, as it's in the intestine, even though it's in the intestine, it's producing metabolites, as you'll see, whose job is to help turn on those antimicrobial peptides. It's to help regulate the immune system so it doesn't over-respond. Helps in terms of actually the levels of antibody that we have in these animals and their susceptibility to infection. All these things are affected by that microbiome that's in the gut or in the respiratory system, right? So one of the groups that's done a lot of work in terms of understanding microbiome in particular in ruminants has been at the University of Alberta, and this is one of their review papers. And so what we're looking at a young calf here. So this is from birth to 12 weeks of age, and we're looking at the mixture of microorganisms that they have. Okay, so you can see as they get older, that things begin to change. So here looking at what's in the small intestine, here's what's in the feces. So they change over time as diet changes. They change. The rumen, you can see that change here. Now, the other interesting thing that these guys showed is that, interestingly enough, so we talk about colostrum and the importance of feeding colostrum to calves. And usually when we talk about that, we think about it in terms of the immune factors, like immunoglobulin and some of the cytokines that I mentioned earlier are important for immune development. But one of the other interesting things that these guys found was that, in fact, if you feed calves colostrum, that their microbiome will be 2 to 3 logs higher than calves that don't get colostrum. So their microbiome develops differently as well. And again, if that microbiome influences immune status, that's another thing in terms of the importance of colostrum in terms of being able to do that. And we think that's really related to actually some smaller molecular weight molecules. There was a good speaker at the Lellman conference that I was at in Florida who actually spoke on this, and it was really quite interesting as they've looked at what the factors are in colostrum that might drive improvement in terms of microbiome health. So if you look then at microbiome in terms of what it could be producing, and I put this up here, and I know this is busy, but I want to just point out to you, so here are these commensal bacteria. So here's these good guys producing factors then that are in particular, and one of the things that has been characterized very nicely is butyrate. So we think about butyrate as being a volatile fatty acid. We think about it in terms of being an energy source, but I'm here to tell you that, in fact, we know that it's a major immune regulator. Butyrate is a major immune regulator. Butyrate itself either can affect these epithelial cells and cause them to produce factors, and that's what you see right here, factors that will turn on these regulatory cells. Because again, what I'm trying to do is I'm trying to keep the immune system from being overreactive, and that's what they're showing here. So here's these regulatory cells. If they're present, that's going to keep all those bad guys or all the inflammatory cells from coming in as fast as they normally do and help keep that system under control. So you see butyrate doing that, one, so by actually affecting these epithelial cells. Another, when it's absorbed, it affects those cells that you saw stick their little process up and help make them produce factors that keep this under control. And then it also affects in terms of other things and just in terms of keeping, again, the immune system from overreacting. And that's what you see all of these things here doing, trying to keep the immune system under control from that standpoint. Again, so butyrate does that, but there's other products that do that. And we know that there's other products, parts of bacteria, that increase mucus formation. Or there's parts of bacteria that increase the production of these antimicrobial peptides. And the other thing that you see here, you see this something called IL-10. That's a cytokine that when it's produced increases IgA. So again, if some of these products that are produced, so either components of the bacteria or the microorganisms or metabolites that they produce will have these effects in terms of immune regulation, and that's really important. And then the other thing, that the term that you see right here is this idea of what's called gut immune homeostasis, the idea that you keep everything kind of in balance. You want to have some inflammatory response, you just don't want a lot, okay? So that's the term that we use is homeostasis. And again, you can see it's driven, in this case, as shown in this slide, actually by what's going on in the lumen itself, okay, in terms of the products that are being produced and the actual bacteria that are there. And this is just another blow up of that, and that's looking at, interestingly, a lot of the work that's been done in the human in the monogastric model has actually looked at clostridials. So usually we think about clostridials, particularly perfringens, as being a bad guy. We talk about Clostridium in the gut, and we think about that's a bad deal. But in fact, most of the Clostridium are good because they digest fiber and they produce short-chain fatty acids or they produce butyrate. And again, here you see, again, that direct effect in terms of having an effect on these cells, which then produce one of these cytokines called TGF-beta, or you see the direct effect here, and again, affecting these regulatory cells. So one is we suppress this over-robust immune response and also improve the production of IgA, because again, that helping enhance that barrier. So we're doing two things here. We're regulating the gut and at the same time enhancing the barrier. One more for me, please. Good. Okay, so now, interestingly enough, so we've been talking about the digestive tract, but this is some work that's been done also in Canada by Edward Timson and his collaborators at the University of Calgary. And this is looking at just okay, so what about what happens in the respiratory tract? Okay, so that what they've done is they've actually looked at a number of stressors, and that we see here. So things like weaning, dehorning, castration, commingling animals, buying them in an auction barn, looking at the effect that they have then on the microbiota of the respiratory tract. Okay, so we've talked about the gut. Now we're talking about the respiratory tract, and the same things hold. And what happens is that you get a change in it. And when you get a change in that microbiota, that's called dysbiosis, because it means it changes, and changes in a way that's not useful to the animal. And so what happens in the respiratory tract in particular is you see colonization and overgrowth of pathogens that are already there. So Mycoplasma bovis, Mannheimia hemolytica, Pasteurella multocida, bugs that are already there, but because now you've changed by these stressors, changed the equation, they now get a chance to overgrow and they actually be able to move down into the lung. And then again, you see additional stressors here. So you see dehydration, nutritional deficiencies, temperature fluctuations, dust affecting how the lung can then clear these out. And of course, that impairs that, and what do you end up with? Bacterial pneumonia. So these things all work together, okay, in terms of, in this case, in a negative way, in terms of enhancing disease. So by having these stresses in the first place, it changes the dynamics of what the bugs are here, and then at the same time, I got additional stresses going on, making it difficult for them to be cleared, and now I get bacterial pneumonia and bovine respiratory disease. All right, so again, this concept of dysbiosis is shown nicely in this slide. Okay, so if you look on the left, what you'll see then is you'll see we have lots of, so the purple ones are the good guys, they're the commensals, lots of them there. They're enhancing, again, by their presence and the products they're producing, enhancing that kill zone. So you see that kill zone right here. So there's the mucus. You see antibody in there. That's a little look like Y's, and then the antimicrobial peptides is the green guys. And again, enhancing that kill zone and keeping everything away. Now, let's say we wean that animal, new diet change. You put that animal on a truck. We ship it from western South Dakota to the panhandle of Texas. And now we look at what does the gut of that animal look like now? Well, we've depleted that microbiota, and when we do that, that completely changes then what can happen. Okay, so now we see, yeah, there's some mucus there, but we don't have very few commensals, and we don't have that anti-inflammatory things going on, and we don't have much going on in terms of the kill zone. So there's no antimicrobial peptides, there's no antibody. Now the pathogens have more opportunity then to enter, cause inflammation, and then cause, and worst case scenario, disease, but probably in most cases, decreased growth and production. Because I've got to put energy into this to now try to fix this, which I would rather be putting that energy into growth and development. So when we think about this microflora as nutritionists, to me it's not just feeding the animal, but it's feeding these microbes. So the idea that what we've seen is when we have more fiber in a diet, guess what it does to the microbiome? It increases the microbiome in terms of the good guys that are present. And of course, we know there's things that have to do like things like production of vitamins and other things that they produce. That's obvious. But then there's other things, interestingly enough, as they've looked at this in people, is that you can actually look at some people that are obese, and you look at their microbiome, and you find that they have bacteria that are quote unquote more efficient in terms of being able actually to synthesize different products. And there's been a couple of interesting studies where they'll take the same line of mice, and they'll take the microbiome from a person that's obese and a person that's thin and put it in the mice, and you can make that mouse obese. Okay, so again, the microbiome itself, the efficiency of the bugs themselves can also have an effect. And we know there's many examples. Rheumatoid arthritis. There's a certain genetic predisposition in people, but when you look at their microbiome, they have more of an inflammatory bacteria in their gut than people that don't. Okay, so again, this interaction with microbiome is really important. All right, so we've talked about the barrier. We've been talking about what does it do to keep that barrier intact. Again, we want to have a good kill zone. We want to have an anti-inflammatory response going as much as possible. Then we talked about this innate immune response, because remember, it's there. What's its job to do? Its job is to sense what's going on. Okay, so it's again, the troops are called to battle. It's to look for injury and infection. The key cell is something called, again, is a macrophage who's on patrol. He's always looking to see what's going on. And again, their job is then to recruit other troops into the situation. And as I told you before, if I don't have an innate immune response, I will not get a vaccine response. Okay, so again, it's very essential that way. All right, so the driver, as I said, is this cell right here called the activated macrophage. And let's just imagine for this example that I'm talking about a vaccine. Okay, so I've given a vaccine, so here's my vaccine. This macrophage, again, being in the sensing mode, senses that there's something there, and then it sends out these inflammatory cytokines, okay, which are the signals that say, hey, I need more cells in here. And again, if it's for a vaccine, what happens, it sends them at sort of at 3 levels. So the first level, so if I give a vaccine and I look at where I give that vaccine locally, what do I see? I see a swelling, okay? And that's because of these cytokines that are right here that I produced, and they're bringing cells into that area, okay? So that's the first thing. The second thing I can't see, but is absolutely essential, is that these cytokines then affect the regional lymph nodes that are close by the injection site. And it turns out for vaccines that regional lymph node is the key thing I need to activate to get a good immune response. So when people talk about lymph nodes swelling, in this case it's important that they do, because I'm going to get a better vaccine response. Okay, so that's kind of a regional response that occurs from the activation of these macrophages. And then the third one, which I'll get to a little bit later, is the systemic response that occurs. And again, systemic response is where we see the things that typically, following vaccination, that the producer's not very happy with, okay? So I see animals go off feed. I see animals that are listless. I see animals with a droopy ear. I see animals with sweats. But all those things, again, tell me that I've engaged the immune system and I got its attention. Now, I mentioned clear at the beginning this idea, this interaction that occurs between the immune system, the nervous system, and in this case, the innate immune system. So here we see, again, so these are the bugs that are out in the gut, in the lumen, and their presence is actually, with their products, will stimulate both neurons, okay, and macrophages. And what you see is, in fact, the neurons, in this case, after they're stimulated, can actually turn the macrophages on, and here you see norepinephrine. So this network of the nervous system and the immune system is very tight, as tied to the gut. So that idea, when you've had, I don't know how many people ate their whole steak last night, but if this morning you said, I have a bad gut, and I'm feeling that, this is where you're feeling it, okay? It really happens, okay? In other words, it's your guts talking to you, okay? And because it turns out the gut's one of the most innervated parts of our body, but that innervation isn't just about going to the brain, it's going to the immune system as well. Okay, so being able to, again, you can see what it says here is that actually, again, the idea that bacteria are picking up on these signals that come from the nervous system, to me, is just amazing. So again, how do we minimize stress to not get those things turned on? Because bacteria are picking up on those and taking advantage of that. All right, so as we said when I was talking about the barrier, the key thing with these epithelial cells is wanting to keep them anti-inflammatory. Because when these pro-inflammatory cytokines come along, it again causes some big problems, and particularly one of them called tumor necrosis factor. Okay, now how many people every night on TV see an ad for Humira or any of these other antibodies that are for rheumatoid arthritis? Okay, we all see those, right? That's all directed against this. All those antibodies are directed against tumor necrosis factor. Okay, because tumor necrosis factor, when we talk about an inflammation in people as well, is the problem. Okay? So when you see all those commercials that we see about things with rheumatoid arthritis are all directed against this one cytokine, tumor necrosis factor. Okay? And it is, as I said, epithelial cell enemy number 1. And we think it relates to the fact that, again, it breaks down tight junctions. Okay, so for our discussion today is the fact that it has on tight junctions. And if we break down tight junctions, then one of the things that we're worried about then is leaky gut. And this is one of Lance Baumgart's slides. Okay, so the idea then that and again, I'm a believer in this, in his hypothesis that leaky gut explains the negative consequences of heat stress and off-feed events in farm animals, okay, regardless of the species that you look at, okay? And again, tumor necrosis factor, as you're going to see, is when the major factor responsible for leaky gut, okay? So leaky gut then affects a number of, obviously, a number of other processes that we'll talk about in just a little bit. But again, the idea that we're affecting in a negative way, particularly the outcome in terms of production. So we're affecting lean and growth, we're affecting milk production with leaky gut because, again, we are putting energy into a different part of the system than we want for growth and lean. All right, so this is what tumor necrosis factor does. And this is the interesting thing about this is it doesn't matter whether the tumor necrosis factor is produced locally, which is what I'm showing you here, or let's say it was in the respiratory tract, or vice versa. Say, so what we're seeing here is here's these macrophages producing this tumor necrosis factor, and the tumor necrosis factor affects a kinase that then breaks down tight junctions. So now I get leaky gut. Okay, it's immune regulated. Okay, so it's these cells right here, and again, you see what we're doing here is if we can shut this down, if we can shut this pathway down, and that's what it's showing here, having these regulatory cells, we will shut this pathway down. And so that's why what I talked about before fits right into this, because we're trying to keep the innate immune system. We want it to be responsive. We just don't want it to be over-responsive. So in this case, we're causing the breakdown of tight junctions and then leaky gut. All right, so now the consequence of that is, okay, so if I have leaky gut and I have endotoxin coming across, it invokes an inflammatory response. So we've already talked about, I showed you previously that local response. So I talked about the fact that, when I gave a vaccine, I have that local response where I can see where I gave the vaccine, or then I have a regional response in the lymph node. But what happens as I have that response increases or it goes systemic, I get the systemic response. And so again, if it was a vaccine, one of the things that I want it to do, which is why it's important, is I want it to stimulate bone marrow so I can get more white blood cells out there so my vaccine works better. So that's why it's essential that I have an innate immune response for a vaccine. Now, to do that though, it's not just one only hit has a one hit. Unfortunately, those same cytokines, when they hit the brain, are the things that cause listlessness, sweats, and animals to go off feed, okay? So depending on where they go, they do different things, okay? So when they go to the brain, sickness behavior, that's what we see, okay? And that's why, because these inflammatory cytokines have an effect there. Now, the third organ they go to though, and that's again, when you talk about leaky gut, it's almost a direct conduit to it, is the liver, okay? All right, now the liver is an interesting organ, okay? So what it does is when it's turned on, you want to hit it for me? It turns on, when it is engaged to be an immune organ, it produces acute phase proteins, okay? And so acute phase proteins are great in terms of a response, but in terms of metabolism and energy, they're a big cost because most of these acute phase proteins are the result of catabolism, and they're not made very efficiently, and so the liver, instead of being a metabolism machine, becomes an immune organ. And the thing that you need to remember is this. The liver is the first immune organ. So when it sees inflammation, it thinks it should be an immune organ again, okay? And how does it see inflammation? Well, if I've got leaky gut and I got endotoxin and I got the production of those inflammatory cytokines, they hit the liver. The liver says, oh, I've got to be an immune organ again. And the thing is, it sucks at that compared to being a metabolism machine that I need for growth and lean, okay? So again, trying to minimize that inflammatory response or leaky gut to try to minimize the engagement of the liver as an inflammatory organ is important if we're looking at trying to be successful in terms of being more efficient with the animal production. There we go. Okay. Now what you're going to see here, one of the cells that's involved is neutrophils. And what you'll see here, and this is the problem when you activate the innate immune system. So what we've got here, these are actually fungi. So these neutrophils are trying to eliminate it, okay? That's their job. Okay, so what you're going to see they're going to do, they're going to work on that, but I want you to keep your eye on this one right here, because this is something that neutrophils unfortunately also do. They do something called net formation, okay? And so what you see, they blow up like an atomic bomb, okay? It does a nice job on the pathogen, but what does it do to the surrounding tissue? Okay? It causes collateral damage, okay? Causes collateral damage, and so then what we have is, guess what? Now we got to fix that, okay? And we know that if you look at the lung, this is a lung. Do you know what almost everything that you see here is? Acute phase proteins that were overproduced. How many of us have ever seen, have looked at a steer that was normal the night before and came back and saw a lung like that? The immune system went over the top. Okay, it over-responded. Okay, so what you're seeing there is, yeah, was Mannheimia there? Yeah, but Mannheimia sucked those neutrophils in, and what did? They did what we just saw, they blew up. Okay, and then we had other acute phase proteins that came in as well. Okay, so now we had the big movie, okay? So we'll see how the volume is here. So this is going to show you actually in the gut, but just review everything that we did.
The human gut can be the scene for devastating conditions such as inflammatory bowel disease, which arises through an improperly controlled immune response. The gut is often the body's first point of contact with microbes. Every mouthful of food is accompanied by a cargo of microorganisms that go on to encounter the mucosa, the innermost layer of the gut. Most microbes are destroyed by the harsh acidic environment in the stomach, but a hardy few make it through to the intestine. The intestinal surface is covered with finger-like protrusions called villi, whose primary function is the absorption of nutrients. However, these structures and the underlying tissues also host the body's largest population of immune cells. Scattered along the intestinal mucosa are dome-like structures called Peyer's patches. These are enriched in lymphoid tissue, making them key sites for coordinating immune responses to pathogens whilst promoting tolerance to harmless microbes and food. The villi contain a network of blood vessels to transport nutrients from food to the rest of the body. Lymphatics from both the Peyer's patches and villi drain into the mesenteric lymph node. Within the villi is a network of loose connective tissue called the lamina propria, and at the base of the villi are the crypts, which host stem cells that replenish the epithelium. Finally, the epithelium, together with its thick overlying mucus, forms an important barrier against microbial invasion. Embedded within the matrix of the Peyer's patch is a mix of immune cells including T and B lymphocytes, macrophages, and dendritic cells. A key function of the Peyer's patch is the sampling of antigens, in this case mostly bacteria and bits of food. To facilitate this, the Peyer's patch has a much thinner mucus layer as well as specialized phagocytic cells called M cells, which can transport material across the epithelial barrier via a process called transcytosis. Finally, dendritic cells are able to extend dendrites between epithelial cells to sample antigens that are then broken down and used for presenting to lymphocytes. Sampling antigens in this way typically results in so-called tolerogenic activation, where the immune system initiates an anti-inflammatory response. With their cargo of antigen, these dendritic cells then traffic to the T cell zones of the Peyer's patch. Upon encounter with specific T cells, the dendritic cells convert them into an immunomodulatory cell called a regulatory T cell, or Treg. Defects in the function of these cells are associated with inflammatory bowel disease in both animals and humans. These Tregs then migrate to the lamina propria of the villi via the lymphatics. Here, the Tregs secrete a molecule called IL-10, which exerts a suppressive action on immune cells within the lamina propria and upon the epithelial layer itself. IL-10 is therefore critical in maintaining immune quiescence and preventing unnecessary inflammation. However, a breakdown in this process of immune homeostasis results in gut pathology, and when this occurs over a prolonged period and in an uncontrolled manner, it can lead to inflammatory bowel disease. Chemical, mechanical, or pathogen-triggered barrier disruption, coupled with particular genetic susceptibilities, may all combine to set off inflammation. Epithelium coming into contact with bacteria is activated, leading to bacterial influx. Alarm molecules released by the epithelium activate immune cells, and Tregs in the vicinity scale down their IL-10 secretion to enable an immune response to proceed. Dendritic cells are also activated by this environment and start to release key inflammatory molecules such as IL-6, IL-12, and IL-23.
These are all pro-inflammatory.
Effector T cells also appear on the scene, and these coordinate an escalation of the immune response by secreting their own inflammatory molecules, tumor necrosis factor alpha, interferon gamma, and IL-17. Soon after the effector T cells have arrived, a voracious phagocyte called a neutrophil is recruited. Neutrophils are critical for the clearance of bacteria. One weapon in the neutrophil armory is the ability to undergo a dramatic form of self-destruction called netosis. This leaves behind a jumble of DNA saturated with enzymes called a neutrophil extracellular trap. Although this can effectively destroy bacterial invaders and plug any breaches in the epithelial wall, it also causes collateral damage to tissues. Slowly the tide begins to turn and the bacterial invasion is repulsed. Any remaining neutrophils die off via apoptosis, a non-inflammatory form of cell death, and are cleared by macrophages. Epithelial integrity is restored by replacement of any damaged cells with new ones from the intestinal crypts. Finally, Tregs are recruited once again to calm the immune response. Targeting the molecules involved in gut pathology is leading to effective therapies for inflammatory bowel disease.
So if you want to see this again, this is actually on YouTube. So just put nature and inflammation in gut and you'll be able to get the movie off of YouTube. But I think I love this movie because it shows the whole idea about when you have damage and then what you have to do to repair that damage, and then there's a cost to that. So those are things that we don't quote unquote see in terms of outright disease, but what does it do to the productivity of that animal? To me, that I really love that movie for that because it really shows that very nicely. But again, the same thing happens in the lung. Now the beauty of the gut, though, at least, is that there's a passageway for that material to go sort of at least an output for it. For the lung, there's nothing. That's the problem with the lung, is that when you have damage in the lung, one, it doesn't regenerate, and two, there's no place to move any products. Okay, so let's just talk a little bit about heat stress and gut health. And again, this is from some of the Lance Baumgard stuff. So again, one of the interesting things when you think about with heat stress in gut health is that since cattle in particular don't sweat, they have to move their blood to the skin and the extremities, then shorting the gut. In other words, decreasing the blood flow to the gut, therefore decreasing the ability of the cells to repair themselves and do that. So again, that vascular constriction that occurs in the intestinal tract is going to again increase the chances that we'll see leaky gut in those animals. In this case, because the fact that the gut it's difficult for the gut to maintain its health under the normal physiological situation. And then we also have the reduced nutrient intake that you have just because of heat stress in terms of increasing the osmolarity. So again, having some negative effect on that response. So the thing then that this can lead to, so again, if we talk about having leaky gut and having a large inflammatory response, if the response is large enough, we can undergo something that's called a cytokine storm. And so these storms, again, are the same things that we talked about in terms of the innate immune response just at a really high level. And we know that, in fact, if you take IL-1 and I give that to an animal IV, that, in fact, I'm going to have temperatures of 104 to 106 without any infection, and that by themselves, cytokines can actually induce respiratory disease. So they can induce, this has been well documented in people, acute lung injury, and then a more kind of a chronic form that's called acute respiratory disease syndrome. It looks to me a little bit more like AIP, but
the interesting thing is, in particular, we see this in dairies with new heifers. They'll bring new heifers in, and they'll bring them in that first week after they calf, and they'll have these sort of fever of unknown origin. And so the response, at least as veterinarians, has often been, well, it's got to be an infectious disease because that's why I've got a fever. And the most likely thing, because I'm seeing respiratory signs that aren't real severe, is BRSV. But if you look at some of the work that's been done, particularly at Kansas State by Barry Bradford, they've actually looked at, and this is in transition cows, actually putting those animals on non-steroidals and actually being able to show that they can decrease mastitis, decrease botrytis, and decrease respiratory disease in those cattle early in that transition period just by controlling inflammation. So again, the cytokine storm to me, and again, you know, I've talked of feed yards in Canada where meloxicam can be used, in cattle, and as we begin to use it in the U.S., where they actually on arrival, rather than using metaphylaxis with antibiotics, they use metaphylaxis with anti-steroid or non-steroidals to try to manage inflammation, because these cattle again have undergone a number of obvious stressors that could increase inflammation. And so this just shows you, back up to this one, again, the idea that with the this acute form that we can again see with these cytokine storms, that actually if I showed you a mannheimia where they have lots of neutrophils, that's pretty much what this looks like. We have lots of neutrophils that have infiltrated into the air spaces. But the more interesting one to me is this chronic form that they see. And that's where you actually see thickening of these airways, which reminds me a lot of what we see with AIP. Okay, so again, things that we see, and I would contend, with this next slide, talk to you a little bit about that as well, because another key part of inflammation is obesity. Okay, so we've learned that in people, and what we see, particularly as we look at in finishing cattle and look at heifers and AIP, the idea that again that we see AIP, which is continually not, we've tried to put an origin on it. And I'm going to contend, I think there's a major inflammatory effect to that. And let me just explain to you what's going on there. So if we look at adipose tissue, so whether we're talking about an overconditioned weaned calf, fat cattle, or transition cow, so these are animals that have a fair amount of fat already laid down. It turns out that the cells that make fat, the adipocytes, when they're active, whether they're actually taking up fat or making fat, they're producing IL-6. And IL-6 is one of those inflammatory cytokines that we talked about earlier. And what that does, among other things, is it recruits macrophages into the fat. So we've got a fair amount of macrophages in the fat. Okay, now I have, here's my gut, here's my microbiome. So now I have, so again, often what we see, so if we talk about AIP, is that we'll see something occurs within a week or so after we've had a change in terms of a ration in terms of what we're doing, in terms of whatever phase that they're in. But if we change, or there's been a heat stress event or something that changes this microbiome. So if we change that microbiome, so we end up with, again, switching from getting more gram negatives, and those gram negatives do what they're supposed to do. They live and they die, but when they die, gram negatives release LPS, and LPS is readily absorbed across in those animals. Again, the LPS then, when it hits these macrophages that have already been activated, just make this thing turn into a very large storm, okay? In the transition cow, it's even a bigger deal because I've got a lot of other things going on as well. But the idea that adipose tissue is also an immune organ, in this case, unfortunately, an inflammatory immune organ, I think we need to be aware of as well in terms of how can we manage that as well. And so again, back to this idea of leaky gut and this permeability. Again, I think we need to think about it in terms of how it can again contribute to the cytokine storms, how it contribute to, in that case, a cytokine storm is an acute disease issue, but probably more times than not, really what we're dealing with are things that are low grade, but certainly affect the ability of that animal to produce. So again, and these are the bad guys. And again, turning on, particularly what they do to the liver, I think is something that we need to be pretty aware of. And again, these are the behaviors that we're going to associate with activation of the innate immune system. Okay, so listlessness, as I said, inaptence, sweats, decreased food conversion, decreased gain. If we're talking about dairy cows, milk production, mastitis, and anything that you're talking about, increased bovine respiratory disease. And again, and it goes back to this, again, the production of LPS and from leaky gut, then again, as we've talked a lot about what it does to the macrophage, we've talked about what it does to the liver, and then in terms of what that can do in terms of actually effect on the muscle. And then we've talked about the effect on the brain in terms of what happens in terms of those lethargy, fever, and impotence that can occur. So again, how these things are all can be tied together, and again, back to episodes of leaky gut. There we go. Okay, and then again, when we have that inflammatory response that's over the top, then again we see this tissue damage, and again that tissue damage has got to be repaired. And in the case of the lung, it can't be repaired. Okay, it's not regenerative in terms of the lung. So that's again something that we need to be aware of. So again, when it comes to innate immunity, then again there's this balancing act that we want to have. The pro-inflammatory response has got to be there because we need it. Is that alarm system? But we know that too much of it can result in disease. And again, it's essential that we have it in terms of our vaccine responses. And on the other side, the anti-inflammatory response, we talked about how that's key in the gut to maintain that gut homeostasis, the production of IgA. But we know that in people, okay, that if we have too much anti-inflammatory response, what do we see there? We see allergy and we see asthma. Okay, so again, all these things they need to be a balance. And again, they work together. So it's not like one against the other, but in fact that they work together. And again, when you look at things that have been done to deal with the innate immune system in terms of management, I mentioned non-steroidals. There was a cytokine that was actually out there to make more neutrophils. It's off the market now, and Rister, used in dairy cows for mastitis. And then we have immunomodulators, such as this is a bacterial cell wall, and then Zelnate from Bayer, which again is a immunomodulator that helps in terms of being able to cause, in this case, macrophages from over-responding. But I think the big opportunity is here. In terms of because if we can manage gut health and be able to again keep that inflammatory response down to a minimum, then these become afterthoughts. Okay, they become afterthoughts because we're ahead of the game. Okay, we're ahead of the game. We're in the preventive mode rather than defensive mode. Okay, so you've seen this for the last time now, and we're going to talk about the adaptive immune response. So again, we've alluded to this a little bit. The last few slides are about that, and that is if we go and look at the mucosal immune response. One of the things that we look at is actually using vaccines that are delivered that way. And again, so what they'll do is here's a nasal cavity. They affect the regional lymph nodes that are there, and then they go ahead and then produce antibodies, and there's T cells there waiting for that response. And again, that's typically what we see. We know, interestingly enough, that there are parental vaccines with different kinds of adjuvants that can also give us that kind of response. So you don't just need to have an intranasal vaccine to actually get a good response against, for example, a respiratory or a gut pathogen, and you're aware of that because people have used those for years. Okay, and so then the thing that happens, can back up one, just back me up one, okay, is that when the immune system is activated then, the adaptive immune response, what it will do is that it can actually decide, do I want that adaptive response to be mainly antibody, or do I want mainly to be memory and T cells? And typically what we want is we want a little bit of both. And that's what I'm showing here, is the idea that, again, so you saw those dendritic cells that are up there, reaching up there, interacting then with T cells, and here you see predominantly a memory response. Okay, and again, we talked about it in the gut. What we want is certainly we want a strong antibody response so we can get IgA. But again, we need some memory and we need antibody as well. So typically we want that response to be pretty well balanced with each other. And again, the other interesting thing about when it comes to the mucosa and this adaptive immune response is that we call this the common immune response. So the idea then is if I actually immunize or get an immune response that occurs in the gut, it will also those cells will traffic back up, and then will actually go to other systems as well. So they can go to the respiratory system, they can go to the reproductive system, they can go to the mammary system. So that's something really unique about a mucosal adaptive immunity, is that it has this ability to actually go to other mucosal surfaces as well. All right, so the take home then is we've talked then about a healthy gut. And so again, looking at the future, which to me is a very bright future of nutraceuticals in terms of being able to, again, be proactive in terms of making this gut as healthy as we can. And again, as we know, there's lots of different options that are out there, but the problem that we've had and Eric and I can certainly tell you about this problem because we've tried to measure this, is how do you measure the kind of response that we're after? How do we measure that? And we've looked at some different experimental systems, but lots of times you'll see people take a peripheral blood sample and try to use that as telling me what's going on in the gut, and I can tell you that that just doesn't work. And that's been one of the problems that we've had, is understanding the mechanism so we can use these strategically based on what we understand how they work. And so again, that's really kind of, as we move forward, something that we really need to, I think, to concentrate on, to understand more about mechanisms, and that will help us in terms of then figure out strategically where would we use different products at different times. And again, my last point here about do we need them all the time? I think it depends, again, on the times of stress and the kinds of stress. And again, as we begin to understand mechanisms, then I think we can be very strategic about what we use. All right, so just to summarize then, so again, we talked about these 3 lines of defense. The mucosa, and I think, I hope that I've emphasized to you the importance of that system. And again, because of the fact that it's anti-inflammatory, the antimicrobial peptides, the kill zone, all of those things very essential. But again, if it's not functioning properly, then we've got issues with leaky gut, and then what does that do in terms of inflammation? And innate immune system is, to me, is kind of a double-edged sword. It is, we need it, it's the reactive defense, but the problem is this issue with collateral damage, because it's the king of that in terms of that. And then finally, acquired immunity gives us the good part of that, again, and typically that's what we have in livestock, fortunately. So we have specific immunity, we have duration of protection. The bad side, and again, this is, again, typically, we see this in older people or in humans in particular, is allergy and chronic inflammation. So that's when it's overused.